Hello, everybody. Welcome to our very exciting seminar today. So today we're delighted to be joined by Professor Barbara Di Ventura. Professor Ventura completed her doctorate in synthetic and systems biology at EMBW Heidelberg before winning not one but two fellowships, I believe, to continue work in systems in computational biology and establishing her group at Heidelberg, which she's later moved to Freiburg. Since starting her own group, she has been highly involved in mentoring several iGEM teams, all of whom have gone on to win a whole list of prizes I don't have time to go through right now. She is also the co-organizer of an international synthetic biology conference called the Designer Bio Conference that I personally attend and enjoy quite a lot, and is chair of a number of committees, including the advisory board for the German Association of Synthetic Biology. Her research span a number of very important areas within molecular and cellular engineering, and she has in particular pioneered the development of a number of important optogenetic tools using protein engineering, and shown that such tools can not only be used for engineering novel functions, but also to understand and study natural biological systems as well. So we are delighted to welcome her today to tell us about her recent work on transcription factor dynamics in mammalian systems.
Yes, thank you very much. And let's see if I manage to allow and share my screen before it works. So it should work as well right now. Okay, and I'm sorry, I don't see any of you, so it's a bit of a funny thing now. I'm looking at my screen and talking to myself. I hope nonetheless to be able to deliver to you the key messages of this talk. And yes, we are going to talk about dynamics. And so as a motivation to you, I guess you all know that gene expression is the reason why we have a cell differentiation. Because cells are contain the same genes, right? The genome is the same, and yet they acquire different capabilities, different morphologies, and so the process that allows this to happen is the process of gene expression, so that not all the genes in this genome are on at any given time. Of course, controlling gene expression, so from the external point of view, as researchers give us a lot of opportunities. On the one end, it can help us really understand the role that a certain protein plays in the cell. And here I'm making a very, very simple scheme to explain this concept. For instance, we are interested in a certain gene X, and we want to know a little bit what this gene X is doing. And then we can say, well, practically it's a similar approach to the knockoff, right? But we don't knock it off, we rather turn it off for a while. And then if we have and we observe a certain phenotype, and if we have a way to then turn it on, we might observe a different phenotype. This could be a growth curve if we are looking at bacteria, for instance. So, of course, this is not at all the whole lot of experiments you should do to understand what this gene is doing, but it might give you already some ideas. On the other hand, controlling gene expression also has a lot of implications for synthetic biology or biotechnology in simple protocols such as protein purification benefit from it, because in some cases, well, we know actually Guy is there, right? He's has done a lot of work on burden, and so we know that cells are stressed, right, when they have to produce a lot of proteins for us, or sometimes the protein of interest is toxic, and so actually it's very good if we can define a certain time point where to start the induction of this gene, because then the cells are happy until that point, or healthier, for instance, and they can use the resources to grow, and then at that time point we start producing the protein that we are interested in. So to do all of this, of course, we need a way to tell the cells, now turn the gene on. So from the, not the natural way, maybe, but we just use some systems that we engineer. So, of course, chemical triggers are powerful, and I'm not going at all to discuss about them. Some are really taken out of nature, right? So that's the way nature itself works, and we have just repurposed these systems for our, in our applications, or other times protein engineering has been involved, and these are very different maybe from what they are in nature. But the major maybe limitation of a chemical trigger is that typically we give it into the medium and it will reach all the cells at the same time, and we get a certain output. And on purpose, I depict it this way because we know that despite the fact that the cells are all genetically identical and they carry the same circuit, they will have different amounts of this reporter, for instance, GFP, because of the noise in gene expression. And again, we're not talking about that too much today, but there is beautiful work done by LINuS and others. So we particularly like light as an external trigger because it has a bunch of interesting properties. It can be really very quickly applied and removed, so it's reversible, and it's very precise from the both temporal and also spatial point of view, spatial point of view, and it's, let's say, relatively cheap. I have to say we never really quantify in terms of money how much, because we keep on claiming light is cheaper. Of course, you have to buy your LED device and so on, but they are indeed very cheap. And if you have to keep on putting some chemical inducers, probably in the long run is more expensive than light when you have bought your equipment at the beginning. So I'm not talking a lot about this, but I thought I would give you just a few slides about that, because this is really the last work that we did in terms of engineering a tool that we hope you guys out there will use very soon in your projects. And the idea was to turn a natural transcriptional regulator from E. coli called AraC from LINuS responsive to light responsive. And to be entirely honest, the concept itself was not novel in terms of doing this kind of swapping game of taking a dimerization domain and swapping it for a light responsive domain. This has been done already in the past from creating this tool called light-on system, where actually they have swapped the dimerization domain of the GAL for transcription factor for the same protein that we also decided to use, which is vivid. And there are many, many others. So maybe there the difference is that we kind of kept really the natural system, so AraC, and we did not take just, let's say, a DNA binding domain from one protein and then turn it into using a transactivation domain from another, for instance, a virus, and use it in mammalian cells. But it was really inside E. coli, this protein exists that does the job, and in response to AraC, can we now turn it into a blue light photosensor? And it actually worked, as you will see in a moment. And as everybody, the people who know me very well, they know that I struggle a lot to give cool names to our tools, because I really personally believe you will be more inclined to use it if it has a nice name that sounds good. And of course, we once we decided to call it BLADE for blue light inducible dimerization of AraC in E. coli, we immediately thought that we had to do something cool with it, and we performed bacteriography. And I'm not explaining to you how it works. It is actually relatively simple to understand to such an audience like you, where we just shine light through a photomask on a plate, and then if the photomask has such an image, you will reproduce it. I mean, if your tool is good enough to do so. So we were very happy to see that we can reach a very beautiful, very good resolution with this tool. What did we do is that we played a bit around with the linker between the DNA binding domain, and that also binds the RNA polymerase domain from AraC and this vivid photosensor, so the one that is in charge of the dimerization. And in this is a very small library we played around with. You see that only when we completely have no linker, so when the linker isn't there, somehow there is probably steric hindrance between these two domains, and it just really doesn't manage to work, and is the only one that doesn't work. Everything else works to some degree, and this one here, the number 5, is the least well performing, and this is because it has a very long linker, and we think that then the two dimerization domain, once it's dimerized, still the linker is so large that the two DNA binding domain are not able to be kept in the right position on the DNA to be as effective as when the linker is shorter. But it's a speculation. We didn't do much experiments on that. And what you see to the left is just a bit of a weaker promoter, and to the right a stronger promoter. And when you use a stronger promoter to drive your transcription factor, And this is a constitutive promoter, of course, because we don't want you to do anything else but just add light and control your reporter. You get higher absolute expression of the reporter in the light, but of course at the expense of having a bit more leakiness. And we really quantify the leakiness here with this dashed line. You really see what you would get when you have the reporter with the promoter, but no transcription factor, because there is always a little bit of activity, right, of the RNA polymerase that can bind nonetheless to its -35 and -10 region, even in the absence of the transcription factor. Another point that I want to make is that AraC wild type, it's always better, okay? And here is actually, I think you can make it go even higher by using certain strain that do not metabolize arabinose, right? This brings other problems maybe, and I don't want to discuss them here right now. You can ask me later. But of course, AraC has been tuned by evolution, and so it's just better than our blade. And if we really wanted to push this dynamic range, we would probably need to go also to laboratory evolution, and this is something that we were not so interested in doing, given the fact that in the literature or out there, you know, there are many fantastic tools for gene expression in bacteria that give you really a very high dynamic range, and we thought that's not what we want to push for. What we instead, the most important point I want to give you here is that what we think Blade allows you to do that other tools right now don't allow you to do is to take from your fridge an existing plasmid or an existing strain that is arabinose sensitive, and you can already do optogenetic experiments without any need of cloning. So you just need to co-transform this P-Blade only that we created for you. So it's a plasmid that really expresses only Blade and no gene of interest, because the gene of interest is in your plasmid that you already have. And you can see that you can get also a nice regulation. The fold change is a bit less because of competition, right, with the AraC that now is on the plasmid. So you have some more AraC being made compared to the previous scenario, but it does work. And we think this is a clear advantage. Maybe not for synthetic biologists. I guess you guys love cloning and do, I don't know, like robotics and stuff, a lot of cloning. So for you, probably this is totally laughable. But we were targeting the microbiology community, and we know that maybe they are not so inclined to do cloning maybe as much as we do in synthetic biology. Very quickly, this works also with strains. So if we were lucky to find a strain, for instance, that was nicely made by Casey Juan, and they have swapped at the chromosomal location the normal promoter that drives the gene ROZ that is involved in the rod shape of E. coli, as the name says, and we swapped it with the PBED, so practically the same promoter that we were working with. So we thought, let's take this strain, and we should be able to kind of copy the same type of experiment they did, controlling the shape of E. coli with light instead of arabinose. So what again we had to do is to take the strain and simply transform it with a plasmid, either the P-Blade or an empty plasmid, just in order to have the same resistances to do the experiments properly. And the rest is in the chromosome. And here you see the representative images and the quantification next to it that I hope convinces you that one advantage of light is that you can revert it. So you can really simply switch it off and you don't need to wash anything, right? So if you have an inducer such as arabinose and now you want to kind of go back to the previous phenotype, you need to get rid of it from the cells, and you need to spin the cells down, wash it with fresh medium. And even if you do so, there is not much reversibility you can get sometimes, like we show it here. So if you look at this part here, this is when we have actually, this part here, this is when we have the strain as it is, right, with an empty plasmid. And once we do the induction, yes, they become more rod shaped, so rod Z is being expressed. But when we recover, so we wash and then we wait some hours, so 2 hours to see what happens, they became even more rod shaped. So they committed to this phenotype, if you like, and they don't go back, because even if we washed AraC, arabinose is still inside the cell, still probably bound to AraC, and they just don't go back. While if you have light, you see that the cells become longer, more rod shape, and then they go back to being round. So, and this is the quantification. And of course, we did only 4-hour induction. Bear in mind that the figure from the paper I showed you before, they did way longer, like 12 hours or more. And that's why they become, as you see here, even more rod shape if we had waited for longer. Okay, so this is already more or less what I want to tell you about this first part and then focus on the other part. Yeah, so we want you to use Blade, hopefully as many as possible, and it's for use in E. coli. It doesn't work. Usually I get the question, I already tell you now, no, it doesn't work in mammalian cells, and I can tell you later why, if you like. And yeah, maybe the fold change is not the highest. So if that's what you need, then please go for other tools. But if you instead want to have facility, right, go right away, if you have already a strain, or simply you like the idea and you don't need such a high fold change because you're controlling a protein with a function in E. coli, as we show in the paper, many proteins with functions in E. coli, you don't need much of them to get the function. It is a single component tool. You don't need to use external chromophores because this works with FAD, which is inside the cells, and it is really tight and it gives you a nice homogeneous response, and this I already said. And it's on Agin, so please just get the plasmid there. Okay, now I enter into the second part. This is a project that we started several years ago, so it was definitely challenging from the technical point of view, and we tackled it in parallel in 2 ways, and I'll tell you in a moment why, how, and today I talk to you more about the synthetic biology approach rather than the natural transcription factor approach. And so transcription factors, especially when we go in the eukaryotic world, so they contain usually several domains like the DNA binding domain, the transactivation domain, and they possess their own signals to come inside the nucleus and be exported out of the nucleus, so the NLSs and the NESs. Doesn't have to be so. So sometimes these components come with partners, right? So they bind to other partners and they get this, they get, for instance, the transactivation domain, like in the TED YAP system, or NLS and NESs come from other proteins. Nonetheless, several have already all of them inside a single protein. And what has become clear in the last, more in the last decade, I would say, is that these transcription factors are not just boringly coming inside the nucleus and remaining there, maybe simply then decaying off. So sometimes they do so, but very often they show interesting dynamics. And dynamics for us are defined as the shape of the curve that describes the activity of the transcription factor over time. And here you see in the plot is always written nuclear localization. And the thing is that one could imagine that if a transcription factor localizes in the nucleus, since this is where the genes are, it will be active. I mean, might not be always the case, right? Because it could be that you need a phosphorylation event, something else on top to be active. So in some cases, this is it, right? Localizing into the nucleus is enough. In other cases, maybe not. And so anyway, these are very interesting dynamics. And many beautiful papers have shown that depending on the input that comes on the cell, there will be different dynamics elicited of the same transcription factor, and then also different gene expression programs are started. This would suggest that the promoters can decode this transcription factor dynamics. So the promoters are a way to tell this transcription factor is coming in waves or is coming as a single long pulse, and that's why in one case this gene is activated, in the other not. And we wanted to understand really at the promoter level what determines this ability to decode transcription factor dynamics. And so we decided to go, as I mentioned quickly before, both trying to study a natural transcription factor that turned out to be very challenging, complicated. So I don't have a story on that yet, but there are actually in the meantime really nice papers. One is the one from Jared, who's sitting in the audience. So there are where, of course, the optogenetics in connection with natural transcription factor has been already used and gave a lot of beautiful biological information and knowledge. We took it on the point of view of synthetic biology because we wanted to work with well-characterized parts for which we really know for instance, information about KDs, or K on and K off, and you know, to do then afterwards a mathematical model, that is written there. So we wanted to work with something which should be as orthogonal as possible to the mammalian cells in which we work, to avoid, actually in this case, the complications of an endogenous regulation on this protein, and that it should be, of course, externally controllable in whichever way. And I already revealed to you that the way we want to go for is light as a way to control the transcription factor. And also the promoters are going to be built in a modular way with well-characterized parts, so that we can exchange only one part, for instance, and then observe what is the effect of having changed only that part. So when you work with natural systems, right, and you look at the natural transcripts, there is really a large level of complication, right, inside the cell, which is very nice also to understand. But here we try to do the simplistic approach to understand something about this question. So how did we build this light-inducible synthetic transcription factor? We exploited the properties of proteins to be made of independently folding domains, right, that have been shown to work also when taken out of the context of the full-length protein. And so you can kind of assemble proteins out of bits and pieces. And so we took one part, which is the DNA binding domain from a bacterial protein called LEX-A. We took the transactivation domain, that is 4 times the BP16 virus-derived transactivation domain that works in mammalian cells, and we needed to visualize this transcription factor, and we took mCherry, which is compatible with the blue light illumination of this tool here, which is LINuS. So LINuS is a tool that we developed some years ago, and it's very simple in concept. So you have to imagine you have a photosensor that is the second love domain of a protein from the plant that undergoes a very dramatic conformational change where helices that are at the N and C terminus that are typically folded in the dark state, they become unfolded and start flopping around, if you want. So they are not so tightly bound to the core as in the dark. And people before us had beautifully shown that you could kind of truncate the helix, the C-terminal helix called J-alpha helix, and append the peptide. And this peptide would be, because of this interaction of the helix with the core domain in the dark, unavailable to bind to a cognate domain. And then once the light is on, because of this conformational change is available. And the only thing we did actually is to specialize this concept, and instead of taking a peptide, we took an NLS. And now we did not need anymore to put any exogenous protein that binds to this peptide, because it's the importance that we target. So they are already endogenously present. So the schematic of the concept is here. We expect this transcription factor to be mostly cytoplasmic in the dark. When we shine blue light, due to the exposure of the NLS, it should come in the nucleus. And there, of course, this DNA binding domain should be able to bind to the cognate operator sequence and start transcription. So here are just some images, microscopic images, to show you that it is the case. So this transcription factor is cytoplasmic in the dark and accumulates in the light in the nucleus and then goes back. And please look, I just want to make you notice right away, the accumulation is not very large. So it's not that we get like a blob like what you see really entirely, right, completely nuclear. But that's enough to see an activity, right? So I think that if you maybe just to give you a message here right away, that at least in some cases, right, depends on what you want to do. That could be enough, right? So if you're looking at biological responses, you don't need a 1,000-fold change. Yeah? It will work very well. And now with this tool, we can impose dynamics as we like, and I put as we like in brackets because it is not entirely true, right? So of course we cannot go, for instance, we realized that the fastest pulses that we could do is this 15-15, because it takes some time for the protein to accumulate in the nucleus, and it takes some time for it to go out of the nucleus. And so somehow these dynamics are a little bit dictated by the time, the fastest time it takes to go down. So if we had wanted to do a 1 minute, type of dynamics, we could not have done it. Yeah? So yes, any dynamics, but there are some constraints to it. But to answer biological questions, these kind of timings were very sufficient. So we decided on 3 different dynamics, the so-called what we call sustained dynamics, where we are having the transcription factor coming in the nucleus and staying there for a period of time, or the transcription factor coming in pulses, where what we change is the interval between these pulses. And we also played around with the amplitude, so how much light we give to obtain either a low signal, like a low amplitude and high amplitude. And this we exploited to understand how sensitive the promoters are to the amplitude of the transcription factor. So here is the pipeline that we used. So we created a single plasmid where we have both the synthetic transcription factor driven by a strong CNV promoter, and then our synthetic promoter that I'll show you in a moment a bit more about, and we have the reporter. This is for, well, actually I'll tell you what it is in a moment. And please note that we decided on purpose to work with trans and transfection. I know that sometimes maybe people would say, why not integration and so on? And we actually did on purpose to, again, exploit the different levels that we get when we do trans and transfection, and then directly obtain information about the sensitivity of the promoters to the different amplitudes of this transcription factor without having to really play around with light, because it would just naturally occur that the cells have different levels. Then we generate the dynamics at the microscope, and we track the synthetic transcription factor and the reporter. We do automated quantification of the data and data stratification, so we kind of cluster depending on these amplitudes, and then try to understand what's going on. And the regime, so the way in which we did the light illumination is more or less like this. We've decided, so again, please maybe ask me later, because I think otherwise I speak for too long and I go too much over time. And why we decided on this, there is a reason. So we decided to really keep the cumulative levels of this synthetic transcription factor as similar as possible across dynamics, rather than the time. So we needed to fix somehow, either play around with the amplitude, right? So to have the same cumulative levels, right, fixing the time, but then you have to have different amplitudes, or rather not caring about the time. So these experiments will have different timings, but will be having the similar cumulative levels. And we decided for this one, again, because we think that the promoters are too sensitive possibly to the amplitudes. We have a 5 hours waiting time because unfortunately the reporter that we are looking at in terms of protein needs that long time to mature. So we created a small library of promoters. So they are characterized by a certain number of response elements. They will be this operator sequence bound by the, let's say, protein. Then we have a Tata box. I mean, here I talk only about the Tata box. We tried also the initiator sequence, but that gave us no reporter at all. So probably too weak in the context of our system. So all the data I show you is about the Tata box. There will be a certain distance with which we play around between the last RE and the Tata box. Then we have the transcription start side and the reporter, and we also created a library of 5' UTR, if you want, where we played around also with the length of this distance here and the strength of the Cossack sequence. And here we also played around not only with the number of transcription of the REs, but also their strength and also the strength of the Tata box. And it took a while, but we eventually then set up something that, of course, a lot of people are already doing, that is to visualize also the nascent RNA together with the protein, because this gives you really more the idea of what's happening at the level of transcription, right? So when you have a report that is a protein, there are many other processes in the cascade meantime. And so we look both at the nascent RNA and at the protein. So I don't explain to you the technology we used. Ask me after if you're interested in. Maybe a little trick, I mean, a point that I want to make is that we had the idea to localize the reporter to the plasma membrane with a CAX motif because we expected these levels to be not very high, and we wanted a way to maybe, you know, kind of amplify the signal a bit, having it on the membrane. We would see it better than when it's, you know, made, but then kind of fills the entire cytosol, and then maybe the signal would become too low for us for detection. So that's the trick we used. Okay, then I will talk to you about 4 promoters. And the first promoter, we created more, but these are the 4 I talked to you about. The first promoter has a strong, so 4 times 4 strong responsive elements and a strong strong data box. And when we impose, right, the different dynamics, and we then look at the nascent reporter RNA and reporter nascent RNA and the reporter protein. And one thing you see immediately, right, is this promoter responds all the time, right? So there is definitely RNA being made and the protein is being made. And actually, when we calculate the mean nascent RNA levels per cell per minute, it's just another way of looking at this data. We don't see a difference. So actually, the different dynamics are not eliciting any different response there. At the level of the protein, we see an increase. And I hope I have the time to explain to you that this is somehow, let's call it an artifact of the fact that we thought we were having similar cumulative levels, but we don't have. And we use our model to know that actually these 2 experiments, because of the different in times, right, there is more of the transcription factor. And that is the reason why we have more of the reporter protein, but the RNA level, this is really what tells us whether a promoter responds to dynamics or not. So we would conclude this does not distinguish dynamics and is also insensitive to amplitude. And maybe this is also understood by the fact that it's so strong, right, that it's really kind of immediately activated also when the signal is not so high. These other 2 promoters, P2 and P3, they can distinguish dynamics. And we can see it from this mean nascent RNA levels per cell per minute data. You really see that the dynamics, you know, the pulses are eliciting less. We have less of this RNA per cell per minute. But when we look at the protein level, we don't see this anymore. And this is in connection to what I showed you before. It's just that because these cumulative levels are higher, then somehow then instead of seeing higher level now for the dynamics, we just see that they are the same. But they do distinguish nonetheless dynamics at this level. And the one is actually insensitive to amplitude and one is sensitive to amplitude. And this makes sense considering that the REs are weak in this case. And so you can expect that you can activate more when you have a higher level of the transcription factor. And the most interesting promoter, of course, is a promoter that has both weak REs and weak data box. And that one is actually filtering out, I mean, yeah, let's call it that the filter is out, so it doesn't respond much to the pulses. And we see, by the way, a nice refractory behavior of this promoter here because we are giving light all the time and the transcription factor is in the nucleus, but nonetheless we see a decrease in the RNA reporter levels during the blue phase and the blue light induction. And we also see, of course, this reflected in the amount of protein that is being made. So here now you really see these both at the level of the RNA and at the level of the protein. And despite the fact that for these 2 data sets we get no statistical significance, but for this one we get it, we actually think that this is sensitive to amplitude because it totally makes sense, right? This as weak REs, and as I showed you for the one before, this makes it sensitive to transcription factor and amplitude, and we think that this is just the fact that this is a more noisy, is a noisier promoter, and that's why statistically, when we do the statistics, we don't see the error bar is so large that we see non-statistical significance, but actually it is sensitive to amplitude. So we did a model, so we were very interested in making a mathematical model that could make us understand better the system and also make some predictions. And so we created a model that has a 3-state promoter model. So we have the promoter can be in an inactive state where the transcription factor is not bound. Can be active, so the transcription factor is bound to a certain fraction of these REs, and the data binding protein is bound to the data box, and the PSC, so the pre-initiation complex, is assembled. Or it can be in a refractory state where despite the fact that the transcription factor is bound to one or more of these arrays, the PSC still cannot be assembled, and question marks about how that can happen and so on. So it's not super clear, but it is observed that this is happening. And we have, of course, some ideas. And then just all the time you saw the data before, right? You see always the dark points, so the black dots, these are the data, really the experiments, and this line, purple or pink, are the fitting, right, and of the model. And we used only the sustained data set to fit the parameters that we could not, guess from the literature. And then we used these, once we fix these parameters, we use the model to predict what would happen when we have other dynamics. And you see that there is really a very beautiful, fitting, no, fitting is the wrong word. So, I mean, so the model can reproduce the data very well. So this is really predictions, okay? We're not using these data set to fit to find the parameters, only this data set. So what is the key message? So to make it very short, we think that actually when the coupling between the transcription factor binding and the pre-initiation complex assembly is very efficient, then actually any dynamic, so also when the transcription factor kind of transiently comes in the nucleus and binds, this will immediately elicit a response because this event is so efficient, right? But if you instead have an inefficient coupling, then actually the transcription factor needs to bind. So the residing time at the promoter has to be longer, right, in order for it to really start the transcription. And that's how then these promoters might filter out pulses where the transcription factor is too transiently inside in the bound, at least bound to the promoter. So having done that, so we asked ourselves now, if this is the core message, can we now turn promoter P1 that was insensitive to dynamics into one that is sensitive to dynamics? So what we thought of doing is to extend the distance between the RE and the data box, because this I didn't mention, but somehow what is known is that there has to be, so a loop is formed once the transcription factor is bound to the REs, and the general transcription factors, the data binding protein, and the other general transcription factors are binding to the data box. There is a looping event, and then with this looping event, then actually the pre-initiation complex really can be assembled, and the RNA polymerase can be called the promoter. And it is known that this looping efficiency depends also on the distance. So we thought if we extend the distance, we are making this coupling less efficient. And so we did both prediction with our model first, and then we constructed physically this new promoter, and we tested it with experiments. And indeed, we see this. We see that both the model would predict that this promoter now senses less, the 1530, for instance, dynamics, and in the experiments, we see even a more dramatic, I have to say, difference in the amount of reported protein that is being made. Then afterwards, we also thought of playing around with this translation initiation. Of course, the reading of the dynamics is done by the promoter, right? So I'm not saying that the translation has to do with that, but it has to do with the kind of amplifying. It can amplify and help, if you want, this decoding that is done at the promoter level. And so what we did is we constructed an extremely small library of only 3 variants. In one case, we have so first by the way, so these are technical details, whatever, we shortened the distance here in order to be able to see something, because when we kept the same distance we had before, the report was anyway too weak, so we didn't see much. So we first shortened that, and that already gave us an increase in the reporter, and we have the perfect Cossack sequence. Or we introduced a mutation that is known to impair a little bit, so it should become less good, or we have absolutely no Cossack at all. And then we look at this, and we see, by the way, that while in these 2 constructs, it seems to not make the difference, right? Only when you really have no Cossack anymore, so now it's really very inefficient, if you like, in locating, so practically in locating the start code, right, and start the translation, then we actually can obtain this promoter that before was not able to sense dynamic very well at the protein level, so was able to do it at the RNA level, but we didn't see this difference in the protein levels. Now all of a sudden shows a nice difference also here. Okay, now the final thing is that, as a little application again back into the synthetic biology, now we can construct, if you want, circuits where we can drive different gene expression programs only using transcription factor dynamics. So in this case, what we created is a very simple circuit where we have two reporter genes. They are driven by two of these promoters in our library, and you might remember P1 is not sensitive to dynamics, is activated no matter what, if you like, and P4 instead is filtering out or does not respond to the dynamics, so to the pulses. And there is a single synthetic transcription factor here. And in this truth table, I show you what we expect to happen. So of course, when we don't impose any light, so there is no light, no transcription factor in the nucleus, should be no gene expression. So that's somehow, it's still a program, but maybe a bit of a boring one. And if we then instead use pulses, we will get only one of the reporters being made, because that one is not made. But when we have instead the sustained dynamics, we get both. And this is then we of course did the experiments and we measured that, and we see that is exactly what happens. So when we have this is the reporter that is driven by P1, and this is the reporter that is driven by P4, and this reporter, so in venous, is made only in standard sustained synthetic transcription factor dynamics and not under the pulses. Of course, this at the moment is only reporter proteins, they don't do anything, it's a bit boring. But of course, the moment in which you change these for some functional proteins, you can really play around and start creating, biological function, right? So where if you need both proteins to enact for a certain process to occur, you will get it only under these conditions and not under the others. So the take-home message, and I hope I was in time. I don't see anyone. I don't have a clock, so I don't know if I spoke too little. If I spoke too little, I can say way more, but usually I know I take too long, so I'm already happy I managed to arrive to the end of this talk. Take-home message. Remember, BLADE is an optogenetic tool to control gene expression in E. coli. So that's for the microbiologist in the audience. LINuS is an optogenetic tool that allows you to control protein localization in mammalian cells. So yeast has been used, and in several really whole organisms. So we're very happy about that this seems to be really transplantable to different systems. Optogenetics is really a powerful experimental technique that can make you study transcription factor dynamics and the role in the cell in biological processes, and natural transcription factors do display different dynamics depending on the stimulus. And now, I mean, it was, of course, always known that promoters must sense them. And I hope that our little study has helped a little bit understand how this is really happening. So at the level of which promoter elements are needed. And let me tell you here, there was the only study that I can really claim has been our inspiration that was very similar in terms of using not optogenetics, but a microfluidic setup to control a yeast transcription factor to make it go in and out of the nucleus. And they did the modeling. It was beautiful work by the Oshii group. So they actually place a lot of emphasis on nucleosomes as a way to control where the promoter sense dynamics due to this nucleosome being positioned or not. And we actually, I hope that we convince you that, well, yes, probably nucleosomes are one way to decapture, to make this coupling, right, less efficient, but you can do it in other ways. So it's not only playing around with epigenetics, if you like, but there are also other ways that can even be then explored on synthetic circuits that are more easy maybe without these complicated chromatin remodeling systems. And so what is important is that this coupling has to be inefficient, and only like this you get this sensing of different dynamics. And inefficient translation initiation is also enhancing this ability of a promoter to distinguish different transcription factor dynamics. And now we can use this knowledge to create more complex, if you want, synthetic circuits that are controlled by dynamics of transcription factors. This is my group. I want to thank especially Enoch. Here he is hiding almost behind the tree. So he has been the head and mind and everything behind this project. Did everything from the modeling to the experiments. The only thing he didn't do was he contributed to the establishment of this automated image processing that took several years. And he had to do quite a lot of manual quantification to help this deep learning algorithm to work well. And I am, sorry, this is something else. So in case there is a question, I can tell you this. I'm happy to get questions from you. And I guess I need to stop sharing the screen. Otherwise, I don't see anyone. Okay. Am I still sharing? No, it's shared. Okay.
Thank you very much. You are. I think you are still sharing. Actually, I can see myself on your screen. Okay.
I'm still sharing. I'll stop sharing. Yes. Yeah. Okay.
Thank you very much. That was a really beautiful demonstration of how these optogenetic tools, and coupled with modeling and everything else, can give us some really interesting insights. There are a couple of questions in the chat, and I was hoping I could ask the people who are asking the questions to ask their own questions, because they can probably ask them better than me. So, Jared, would you like to start?
Hey, Barbara, how's it going? Questions from the usual suspects.
Good. Okay.
I wrote two questions, and maybe I'll just ask them both because I think I can do it pretty quick. The first one is just that the dynamics of nuclear import and export seemed a little slower than I would have expected from some other stuff. And I was wondering if this is different than what you get when you do LINuS just with mcherry, like if the transcription factor is slowing things down.
Yeah, I think so. Okay, I cannot really tell whether this is the thing. I think that in this, because we do very long experiments, AraC has really went down with the light a lot. Right, and so maybe it has to do with that, right? He really tries to use the illumination that still allows him to see the translocation and so on, but to keep the cells as happy as possible. And so I think that if we are using very different setup than what we were using with just our Nature Communication paper with LINuS only, where we were having higher. So I think one aspect is that, and if we had more light, right, because it's always a balance, right? So we probably don't really illuminate we don't open up really all of the molecules, right? So maybe that's why you always have some more that are remaining in the cytoplasm, of course, and this you see it, of course, from the how you say, the fact that we don't accumulate a lot, but this, of course, is not how fast the single molecule is coming inside, right?
Right, right. That makes sense.
Yeah, maybe though it could be that he has also used a bit of a longer interval between the next pulse, and in that case, then once it is back in the dark state, we would not catch it immediately after, and maybe that allows it to meantime be exported a bit, and this could slow down a little bit.
Yeah, I think dim light, low light activation intensities could probably slow the dynamics down. That makes a lot of sense.
Yeah, because otherwise, I mean, the only thing that I could imagine is that if you have a transcription factor, this could potentially affect, or we saw that in our paper, but more the export part, because being a DNA binding protein, right, it kind of grabs the DNA and it is slowed down, but more for the export than the import, right? So if you talk about the import, there could only be two reasons. There is an anchor, right? There is something that keeps it in the cytoplasm, but we don't know, we can't imagine that because, like say it's bacterial, the transactivation domain is a virus, from a virus, and cherry also from a jellyfish, whatever, so a courier. Yeah. So it's hard to imagine, right? Why, what would be the anchor? And the other thing is the DNLS, right? It has really to do with LINuS and the exposure of DNLS. That would be my...
The other question I had was about the refractory state. So you mentioned it, the original data, and it makes me so excited. I think it's so cool. And it suggests the possibility that you could have a case where repeated pulses would activate repeated transcription, but a sustained input would cause the refractory state. And so you would get higher GFP from pulses than from a sustained input. And I was wondering if you observed that, or if your model predicts that, or if there is some case where that could happen, because that would be a really exciting thing for how dynamics could be interpreted, even without feedback loops, just directly by the promoter.
Yeah, no, so we actually don't see that, right? We don't see that with the pulses you get higher. We never get higher 9. So actually, well, that would be the slide into which I was going when I was too fast. So we really believe this, that if you have only these components that make a promoter, right, and you don't consider feedback in your paper, right, then there is no way that I mean, there is a way, but but it has to be a bit complicated. So to have without another molecule, in order for it to happen, is we imagine at least one scenario is that after a certain threshold of the transcription factor, then the promoter goes into this refractory state, and we envisage the possibility of forming this condensate, right? This drop, this everybody speaks about that, right? And we think that if the transcription factor, when it stays there long enough at a high enough concentration, can form these droplets, and these droplets are not activating but rather repressing because they kick out the components of the transcription machinery, right? Then you get into a repressed state, right? So the transcription factor is there, but nothing is happening. And theoretically, the model tells, yes, you can do it. So we have that. In theory, we show it's possible. And, when we sent the paper the first time, the reviewers were asking, of course, they all went into that. They were super excited about that, but, build it or show it. But I mean, practically with the components we have right now, we can't show this. It's going to be either activated by both or activated by the sustain and not the pulses. And only if we start creating this system where the droplets are formed and so on. And we cite a paper. There is a paper that is the closest that we can get to this concept, where they show something similar. They show droplets that are repressing instead of activating. And now, we hope we kind of restructure the paper a bit by saying, this can happen, and look at this paper, so that they don't kill us by saying construct it. This will be other years of work, right? And eventually, my student has enough.
Thank you very much. Those are great answers.
Yes.
Guy, you had a question, I think?
I think my question was related to that of Jared. I mean, I was asking. It looks like the LINuS system that you built does some form. I mean, it's difficult to say because we didn't explore all the possible pulse width modulation frequencies, but it does some form of band pass filtering or low pass filtering. From what you have just discussed, it seems to be low pass, not band pass. And I was just wondering if there is any possibility of characterizing basically the cutoff frequencies that allows us to, that would give you this distinguishing feature between, the promoter gets activated or not, depending on, the frequency of these pulses.
It's a good point. No, so the thing is, it's doable. It's totally doable, but it's a very, tedious kind of experiment, because we need to always, so let's put it like this. For instance, a longer interval becomes really actually practically impossible, sadly so, because the time it takes for us to do the experiment, right, to keep this cumulative level the same, becomes even longer. Right now we are already talking about 8 hours or more, and in this time the cells are either detaching, they divide, or die or whatever, and we get at the end so few cells that a single experiment, right, even if we run a lot of experiments, we get so few cells out of it that we will need to run really a lot, right? And so in terms of making it faster, we can't go faster than we did, right? So the 15 minutes is the minimum time, right, in order that it really goes out to then go back in. So shorter than that, we cannot go. We can go longer, but then there is a technical challenge of the hours that it takes, right? So, and that's why somehow...
I mean, Jared, you also answered that question. You alluded to the fact that maybe the dynamics, the dynamics would be shorter in the order of minutes, a couple of minutes, 5 minutes, something like that.
Oh, so short, yeah, okay, yeah, that we didn't think of doing.
And there might be a possibility. So if you do pulse width modulation, you can actually sweep. You can change the duty cycle continuously, and that is the equivalent actually of going from continuous illumination to more spaced out illumination in one single go. So it's the equivalent of what we call a sweep in frequency.
Okay, like Jared did in his paper, in this AraC paper. Okay, yeah, we can think about that. We have not we have not gone in that direction. There was a beautiful paper, right, where you kind of mix all of the frequencies, right, and then you go, you were doing like Fourier transform or something like this, not to go into the frequency space. Yeah, yeah, I mean, we can think about it. It's just, again, so we did not go in that direction at the beginning of time, and I don't know if my student want to publish the paper and move on to the next thing. So it's always a bit the sad story, but it can be a continuation and try to do it also this other way. Because for the moment, maybe because this is a synthetic promoter and it's not really, say, the AraC promoter or the P53 promoter, where maybe really knowing which frequencies are, so in such a specific way, which frequencies are, where is the cutoff, right? You see what I mean? So it's true, it is a low band pass, or high actually, I would say, but it doesn't really, it's a low band pass filter, so it filters out, but it's, maybe not so important to know specifically where that is in this specific possibly, right?
Does your mathematical model give you the link that exists between these parameters that you have in your design with the cutoff frequency? Because
that we could use. Yeah, that's a beautiful thing. Yes, that we can use the model. I mean, the point is that you do the model, and maybe then people will ask you to do the experiments, but we can do the model just for us, at least for us to know, because the moment in which we claim something in the paper saying we use the model to do this, then probably they will ask us to also do it. Like when we did this theoretical analysis to show that promoters that sense better pulses compared to sustained signal exist, immediately they ask, build it, right? And I mean, thank you very much. Yeah, so that's a bit the solution. But yeah, it's a point. We'll keep it in mind.
Cheers.
There's one more question in the Q&A, so I wanted to maybe end by asking Yi to ask a question before closing the formal part. See there?
Anyway, I'm reading it in case.
Okay.
Yeah, it's a very simple, because I think in the paper we wrote, so we even explore different intensity. Now I'm so bad always in remembering these numbers, but something like 1 microwatt per square meter or something like this. But so it is absolutely not a high intensity. So it's absolutely achievable with a normal LED. We also just bought, I think, from Amazon. So like nothing super custom made by some complicated microscopy company. So it's actually easy. It doesn't need a lot of light because it comes from a plant, right, that exploits the light of the sun, and so it's not excessive light you need. So definitely you can get a normal LED.
Okay, well, we've come to 5 o'clock, so I think I'm going to close the formal part of the seminar. I want to thank Barbara again for the seminar. It was really very interesting, and I want to thank the